My name is Jean Fan. I'm a fourth-year graduate student in Peter Kharchenko's lab, and I'd like to thank the organizers for giving me this opportunity to tell you a little bit about my work in linking genetic and transcriptional intratumoral heterogeneity at the single-cell level. And of course, thanks to all of you for getting up early for this talk. So just as an overview of my talk, I'll first give a very brief background and motivation for why to take a single-cell approach at this. No doubt I'm kind of preaching to the choir here. But then I'll talk about our work with a targeted integrative approach for dissecting how genetic and transcriptional heterogeneity are interlinked in chronic lymphocytic leukemia. And lastly, I'll talk about an unbiased computational approach with a software called Honey Badger that I've been developing in the lab. So first, just for why it's important to study intratumoral heterogeneity, I think it's been long appreciated that tumors in different patients are very different, but now it's becoming more appreciated that even within the same individual tumor, we might have different subclones, and like within the same tumor, not all the cells are the same. So that's what we call intratumoral heterogeneity. And understanding this intratumoral heterogeneity is very important because we've seen from some of our previous work with Jan Berger and Dan Landau and Catherine Wu's lab that we can observe these clonal evolutions and changes in subclonal proportions over time. So in this here, I guess the laser pointer doesn't show up very great. But you can see that, so this is a particular patient where we have longitudinal data, and in the first initial time point, they had a particular SF3B1 mutation affecting all the tumor cells, but then over time, they had relapses and went on ibrutinib treatment, and unfortunately again relapsed, and then went into salvage therapy. But we can see that the dominant subclones and the mutational frequencies change over time. And by the time they had their final relapse, all the cells had the original SF3B1 mutation, but then they gained additional mutations in UTF1 and PLCG2. And there are actually subclones with different PLCG2 mutations that we were able to see through bulk sequencing. But unfortunately, through bulk sequencing alone, we're not able to resolve whether these mutations affect the same cells or whether they're actually affecting different cells. So for that, we actually have to take a single-cell sequencing approach, which was what we did. So here, each column is a single cell, each row is a different mutation. And as I mentioned before, we saw a lot of these PLCG2 mutations occur at the final relapse time point. And we were able to see through targeted single-cell sequencing that indeed all the cells had a particular SF3B1 mutation, which we knew. But then when we look at these PLCG2 mutations, we see that rather than affecting the same single cells in a kind of branching architecture, they're actually mutually exclusive. So we're able to, through single-cell sequencing, be able to distinguish between these two scenarios. Now, what ideally we would want to do next is, now that we've resolved that these mutations are actually occurring in different single cells, be able to ask what are the transcriptional differences between these cells. And of course, understanding how transcriptional differences are affected by genetic mutations and vice versa is important. Obviously here, through the work of Gad Getz and many others, including my collaborations with Lili Wang and Angela Brooks, a former Brody, now professor at UCSC, and Catherine Wu, we were able to use previous bulk-based approaches to study the effects of transcriptional impact of SF3B1 mutation, which is a very common mutation in chronic lymphocytic leukemia. So for this, SF3B1 is actually a commonly clonal mutation. So then, because it was clonal, we had lots of chronic lymphocytic leukemia samples with mutant SF3B1 clonal, and be able to compare that with chronic lymphocytic leukemia samples without SF3B1 mutation, and then also compare it to bulk normal B cells. And as you can imagine here, each column is a sample. Each row is actually a different alternative splice variant. You can also look at gene expression. But the general takeaway is that by comparing these samples with clonal SF3B1 versus wild-type SF3B1, we're able to see certain splice patterns that are like alternative splice variants that are differentially included in the samples with SF3B1 versus not in the CLL samples with wild-type and also in normal. And then based on that, we can start asking questions, well, what gene sets are these splice variants enriched in? What pathways do they affect? And do more functional follow-up on the specific splice variants. But of course, this all requires lots of samples with and without clonal SF3B1 mutation. If we were to take a single cell approach, let's consider if we had a sample with a subclonal SF3B1 mutation. Ideally, we would like to be able to see lots of single cells with the SF3B1 mutation and lots of single cells from the same tumor without the SF3B1 mutation, and do a similar comparison on the transcriptional level between the cells with and without the mutation within the same tumor subclone, or within the same tumor. But of course, that requires simultaneous assessment of transcriptome and genetics from the same single cells. And even though we do have certain techniques like GNT-seq, you might have heard from Marioni's lab, generally the approaches for doing this simultaneous assessment of DNA and RNA level information from the same single cells is still quite nascent. So actually, in collaboration with Catherine Wu and CZ Zhang and Lili Wang, we developed another integrated, targeted approach for simultaneous assessment of DNA and RNA level information from the same single cells. So here we first, in this project, which is hopefully coming out soon, we're actually presenting it at AACR in Boston, if anyone is going. So in this approach, we first took a targeted DNA sequencing approach just to get a ground truth. We were able to sequence a number of samples and show that in this particular sample, we have, let's say, a clonal ATM mutation and a subclonal mTOR mutation that was mutually exclusive from a subclonal LCP1 mutation. So if we were to reconstruct the tree, we would suspect that ATM mutation maybe occurred early on, and then there's an LCP1 mutation in this subclone and an mTOR mutation in this one. And then after that, we thought, okay, let's just do single-cell RNA-seq and try to infer these mutations directly from single-cell RNA-seq, and that will allow us to look at both the transcriptional changes and the genetic alterations. So using an approach I published on previously called pathway and gene set over dispersion analysis, we were able to look for coordinated aspects of transcriptional heterogeneity within the single cells. So here each column is a single cell. Each row, you can imagine, rather than looking at gene expression, we're looking at coordinated variability within pathways. So you can think of orange as high expression of genes within this particular pathway. We can actually also look at particular genes within each pathway. So what we were really hoping to see from this analysis was, yes, there are aspects of transcriptional heterogeneity, but we were hoping to be able to identify, look at the same mutations like mTOR and LCP1 and see, okay, maybe all the cells with LCP1 mutation are in this group, and all the cells with mTOR mutation are in this group, and that in that way be able to connect the genetic and transcriptional heterogeneity. Unfortunately, what we saw was that because of really sparse coverage, or both sparse and uneven coverage, we weren't exactly able to do that. So here, each column is again a single cell, and each row is a different mutation. And black means mutant, so and gray means no coverage. So just from this alone, you can see most of the mutation sites did not have coverage, so we're not able to call the mutation. When we do see, we do have coverage of the site, ATM is indeed, I guess, clonal based on these two observations, but we really aren't able to resolve which subclonal branch is which. And that really motivated us, because the mutation calling was so sparse from single-cell RNA-seq, we had to take a different targeted approach. So we, I guess in general, I just wanted to make a point that this isn't because our sample was particularly poor or anything. We and others have also seen that monoallelic detection in single-cell RNA-seq is a very common issue. that in general, if your coverage is very low, which is common in RNA-seq, you have a very high chance of detecting only one allele. So if you want to be able to see both alleles and be able to resolve the mutations, you need very high coverages, which is very rare, I guess, in single-cell RNA-seq compared to bulk. So therefore, we decided to take a targeted multiplexed RT-qPCR approach, where we actually, essentially, we sort the single cells into plates, perform cDNA preamplification, and then divide that preamplified library into 2 different assays that were able to detect 96 different targeted gene expression or 48 different somatic mutations. And the way we do that is actually by designing primers for both the mutant and the normal allele. So we can detect expression of the mutant allele and also on the y-axis expression of the normal allele. So for example, for a particular mutation, we would design primers for both alleles. And then in normal B cells, what you would expect is that we would only detect the normal allele and basically none of the mutant allele. And of course, since the 2 primers are only different by 1 nucleotide, you do get this phenomenon of crosstalk, where you get some detection of the mutant allele even in normal samples. So we have to just simply model that as a simple linear regression and assess what is the rate of mutant allele detection as a function of normal allele detection. And then when we look at tumor samples, we can clearly see that there are some cells that are mostly detecting the normal allele, and we don't detect the mutant allele above what's expected. But there are also these groups of cells where we can clearly see expression of the mutant allele, and that, therefore, these are cells with the mutation. And from that, we can just confirm that our targeted single-cell mutation detection matches our expectations from both the bulk cancer cell fraction and also from single-cell DNA. There's a very nice linear correlation here. And from that, we're then able to actually resolve the subclonal structure. So here is our original single-cell DNA approach, and here's the targeted RNA-based mutation inference from RT-qPCR. And indeed, we see a nice clonal ATM mutation and a subclonal LCP1 mutation. So now we're able to resolve the 2 genetic branches. And because we have the library split, we're now able to also assay for targeted transcriptional changes in the same exact single cells. So for this, of course, you have to know a little bit beforehand which genes to pick. You're limited to, I guess, 96 or multiplicates of 96. So we decided to look at genes that we knew to be highly variable based on our single-cell RNA-seq analysis, just to see whether they were coordinatedly variable between the 2 genetic branches. So, and unfortunately, what we ended up seeing was that there really was not much of a difference between the 2 branches in terms of expression of these genes. And okay, at first we thought, okay, maybe our assays aren't that great. But then we were able to see that for genes within this particular branch, which had a known deletion of 13q and 11q, that genes within these genes within the deletion regions were actually expectedly lower in cells with the deletions than without. So yeah, pick, for example, ATM is within this 11q, and we can see that for cells that in the branch that had deletions, the expression of this gene is lower. And again, we just wanted to have a sanity check, so we picked a few housekeeping genes like B2M and PAX5 and show that expression between the 2 genetic branches are the same. So because we were able to see that genes driving aspects of transcriptional heterogeneity did not differ much between the 2 genetic branches, this led us to pursue the idea that maybe these 2 genetic branches are actually phenocopying each other. And Lili Wang was able to do a lot of excellent wet lab work just to confirm that indeed this LCP1 mutation induced certain changes in DNA damage response and cell cycle and proliferation that was consistent with our idea of phenocopying. So lastly, I wanted to...
Question.
Mm-hmm.
What are you plotting with the box plot? You're not plotting typical median quartiles, right?
So this is median and interquartile, yes.
Oh, you are.
Yeah.
What's up with the CUGT one?
Let's see, which one is SUGT1. So that looks like this gene is basically 0 in the cells in this branch, and I guess the median is also 0, but it's more, so these are, I guess, our outliers, and yeah, so upper quartile is a bit higher.
How come you're not using the chromosomal SCD?
Yeah, that's a good question. So SCD requires, so this is targeted RT-qPCR, so we only have 96. SCD requires error models to be built based on, it requires a lot of cells of different, a lot of genes, sorry, of different magnitudes. So if we were to try to build an error model using just 96 genes, it ends up being rather poor. But that's a good question. Yeah, so our approaches are a little bit different here. And also the dropout rates for RT-qPCR are a little better. So it's generally, yeah, not as heavy. Yeah. Okay, for some reason my clicker, I think, is out of power, so I will move over here. So I've shown you that it's really challenging to infer single point mutations from RNA-seq, but as you can see in these branches, we're not restricted to just point mutation information. We also have deletion information. And even though we can't identify subclonal point mutations from single-cell RNA-seq, maybe we can use these, try to identify these focal deletions. And why I believe we're able to do that is because focal deletions affect, rather than affecting one particular point, it affects multiple SNPs within that deletion region in a coordinated manner. So
just for the sake of the audience, which has less biology background, what is a focal deletion?
Focal deletion is, I guess, a small deletion. I guess, yeah. So in the context that I'm speaking, it's a deletion that's not a chromosome arm deletion. Okay. So for example, you can see in this illustration, if we were to look at, I guess traditionally when we're identifying deletions in bulk, you can look at differences in allelic imbalance. So for example, in a deletion region, because you only have one allele, here each column is a heterozygous SNP, and each row is a single cell. So if you were to examine a region that has, in this particular picture, it's a clonal deletion, all the cells have this particular deletion, so you can only get expression from one allele because you don't have the other one. So then you're able to see only expression of one allele, so pure allelic imbalance in one direction. In a neutral region, in theory, ideally, what you would see is like something what you see in bulk. Here, yellow means detection of both alleles. So in theory, in a perfect world, we would have equal detection of both alleles in every single cell. Unfortunately, due to monoallelic detection, what we often see is expression of, in this particular cell, we have expression of one allele, and in this cell, we have expression of the other allele in a pure allelic imbalance manner. However, we can see that within an entire region, you can see different alleles being expressed. So we still know that this cell has both alleles because we can look at across SNPs within this entire region. And generally, we do see coordination within a gene. So for example, in this particular gene, all the SNPs are, you can only see expression of one allele of the gene, which is what we would expect at the single cell level. So in order to, I guess, quantify this observation, we developed HMM integrated Bayesian approach to detect copy number variation changes from single cell RNA-seq data. I call it Honey Badger. So the purpose of Honey Badger is to use a Bayesian hierarchical model that says, given a region and given the allele observations and the allelic imbalance, what is the posterior probability that there is actually a deletion or LOH in this, cell, given our observations. And then the HMM part is to try to identify the regions that are potentially impacted by these deletions to test in the first place. So just a brief overview of the Bayesian model. The general idea is that we're trying to infer the presence of a deletion in a particular cell. So what we're really interested in is this S. So that's kind of our higher order level of the model. But we don't get to observe that. What we actually observe are these SNPs. So we can get counts of both the major allele and the minor allele. And generally, the SNPs are organized within genes. And therefore, genes exhibit monoallelic expression in one particular way or another. This tends to be related to the gene expression magnitude and also generally related to gene expression magnitude, so we can build that into the model. So we have a SNP level of information that's organized within a gene level information, and that was within a cell level organization.
What is B?
B here is the degree of the bias. For example, we expect that 80% of the genes of this particular magnitude will exhibit monoallelic expression. And D is the direction of the bias, whether you're biased towards the major allele or the minor allele. So I think just to draw out what this means practically, for example, if we were to observe coordinated depletion of a particular allele across many genes, we would have a high posterior probability that in this particular cell, we do have a deletion. However, we could also see something like this, where there are 3 SNPs that show coordinated expression of only one allele, so pure allelic imbalance. However, they're all contained within one gene. So therefore, even if we were testing just this region to see if there's a focal deletion, we would get a very low posterior probability simply because within a particular gene, we know there to be high levels of monoallelic detection.
Is that how long have we known that? That within particular genes, if you look at the bulk level, we see sort of some mean measure, but then when you look at individual cells, was it sort of a surprise that that's the case, that each cell might silence one or the other? Yeah,
so actually that's a really great question because it gets to whether this is a biological phenomena or whether it's a purely measurement phenomena. And I think people have published on how there is prevalent monoallelic detection in single-cell RNA-seq, but I think it is a purely measurement phenomena, that it's not biological. And that belief is based on the fact that in RT-qPCR, even when we're measuring the RNA level information, we do see both alleles. But if you were to prepare a library for RNA-seq, even for the same sample, then you can't see both alleles. So it makes me think that it's a issue of the reverse transcription or something about the single-cell RNA-seq library preparation.
Could it be a timing issue that maybe it's stochastic at any time, only one of them is expressing at a high rate?
That's true.
Back and forth?
Yeah, that is possible. So maybe in RT-qPCR, there's, I guess, more, we're capturing a longer time span.
Yeah.
So yeah, actually, that's a good point. I don't know. So I think it'd be interesting for someone to find out.
Question. Is there a difference in genes where both alleles can be detected, like third column versus column?
Yes. in general, this gene I would expect to be much more highly expressed than like this one. Yeah, so yeah, we've shown previously that this monoallelic rate is a function of gene expression magnitude. And that gets back at how this is probably a measurement phenomenon. So in order to identify the regions, so yeah, basically this Bayesian model takes as an input the observed allelic ratios, but then you also need to provide a particular region to test. So in order to identify those regions, we use a hidden Markov model where what's hidden is whether this SNP is a part of a deletion, and what's observed is the pool of minor allele counts and coverage counts. So for this, we actually end up having to pool all the single cells together to run this HMM model on it, because running it on an individual cell creates lots of spurious signals. So the idea is that given a sufficiently homogeneous in silico mini pool of single cells, we can identify regions. Identify regions that are potentially affected by deletions, and then go back and now that we have these regions, test each region individually in each single cell to assess the posterior probability of the deletion in each single cell. So the general workflow is then to essentially take all your single cells, try to cluster them a little bit based on their, to get more homogeneous pools, pool those together, run an HMM on it to identify regions, and then be able to use our hierarchical model to assess the posterior probability of each deletion. So here we found 2 deletions in this model using our HMM, and now we can assess the posterior probability in each single cell, which is each row. And based on that, divide up the cells into 2 genetic branches, and essentially recursively or iteratively apply this method to construct the rest of our phylogenetic tree.
So I think this one, for example, so a very ad hoc, simple way to do this would be to compare across cells the general RNA transcription level of a gene to see if there's a copy variation.
Yeah. So I'll actually be talking about that a little bit. So you're jumping a little bit ahead, but so I guess first I just wanted to mention one thing that as a sanity check, we wanted to see that we could distinguish normal cells from tumor cells using this approach. And we happen to have this is multiple myeloma samples from the same patient, so 2 different samples, but one had really high purity and one had really low purity. So we would expect that most of the cells inferred from this sample would be normal, and most of the cells inferred from this sample would be have lots of deletions, and therefore be tumor. And indeed, when we run honey badger on this, we can these are the regions that get picked up as being affected, and therefore we can then go back and test in each individual cell what is their deletion status. So black here means high probability of deletion, and beige is low probability of deletion. And when we see how this is associated with which sample it came from, indeed, the sample with low purity had mostly normals, and the sample with high purity had mostly tumor cells. And we were also able to show that the deletions that we found were indeed true based on bulk exome-seq and FISH. So yeah, now to get to the comparison with expression-based karyotyping, of course, this idea of inferring genetic level information from a single-cell RNA-seq is such a good idea that we weren't the only ones who had the idea. So previously in Aviv and Brad Bernstein's lab, they've published on this approach called expression-based karyotyping, where based on deviations from what's expected based on a normal reference, you can identify, so here I guess each column is a gene position, but it's smooth, and each row is a different single cell. So for cells with, for example, an amplification, you would expect to see higher expression of genes within that region of amplification compared to a normal. And in regions, so chromosome 10 here is a deletion, so the gene expression is lower than what you would expect from normal. So based on this picture, we can see obviously there are some normal cells, and also there's a clonal chromosome 10 deletion, and also other deletions as well. So just to compare our honey badger approach to this expression-based approach, we essentially were able to run our approach and identify the same deletions on chromosome 10, so 10, 13, and 14. But what's really cool is we were also able to pick up this really tiny focal deletion on chromosome, I think that was 17 or something. And when we look closer at the allelic profiles of these deletions, so this tiny chromosome 17 deletion has the same clonality as the chromosome 10 deletion, which makes us think that it is real, but it just wasn't picked up by the expression-based karyotyping approach because it was so small. And we're able to use simulations to show that our ability to identify correctly and precisely identify SNPs affected by deletions is, of course, a function of the size of the deletion. So for deletions that are like 101 megabases, chromosome level, you have very high accuracy, and also it's a function of clonality. So if the deletion affects all the cells, then we have a much better shot at getting it. And in general, that decreases as clonality decreases and also as the size of the deletion decreases. And similarly, we can assess our performance to distinguish normal cells from mutant cells. And again, this is a function of both the deletion size and the clonality. And when we compare it to the gene expression karyotyping, we can see that for for chromosome arm level or chromosome level deletions, the gene expression karyotyping approach does extremely well. But of course, for very small, like 10 megabase level deletions, the expression-based karyotyping method is substantially less sensitive and precise. And similarly, we can see that for the ability to distinguish normal and tumor cells as well, that the expression-based karyotyping approach, while very powerful for identifying large chromosome arm level alterations, is really struggling when it comes to smaller focal deletions. Just to emphasize another difference between the expression-based karyotyping approach and the allelic-based approach. So we analyzed some data from a publication by Chang et al. And in this publication, they looked at breast cancer samples that they knew to have poor purity and wanted to distinguish tumor from normal. So there was this particular BCO9 sample where they looked at they used expression-based karyotyping to look at to try to infer deletions and alterations and show that it looked mostly normal. However, when we took an allele-based approach, we immediately see that there are some cells that look a lot different than all the others. And what was really cool is then we were able to do whole exome sequencing on the tumor and show that this is a copy-neutral LOH. So the expression-based karyotyping showed that this was a copy-neutral region, which is correct. But the allele-based approach was able to see that even though this was copy-neutral, there was still something strange about the allelic imbalance. And combined, we can then start being able to distinguish whether that this is actually a copy-neutral LOH. So I thought.
Can you expand copy-neutral LOH?
Yeah. So copy-neutral LOH. So normally you get two alleles. Copy-neutral LOH is when you lose one, but then duplicate the other. So you still have two alleles, but they're both one. And typically you expect to see heterozygous SNPs, but now you don't because you have two of the same alleles. So yeah, so that was really cool. And just as a last example, so in collaboration with Peter Park and SGI group, Hyuk Lee, we analyzed this particular progressive multiple myeloma sample where the patient had an initial bone marrow sequence that showed generally copy neutral, except for one deletion on chromosome 13. And then a few months later, this patient came back and had an ascites dissemination, which we were able to sequence and started seeing a lot more deletions. So we were wondering maybe there was a subclone, a small subclone in the initial sample that harbored these mutations that eventually expanded, but we weren't able to see because of the bulk pooling. So we applied honey badger to the second time point and identified a number of clonal deletions. And when we assessed the posterior probability in the ascites dissemination second sample, indeed all of these mutations were clonal. But what's exciting is now, well, we also have single cells from the first time point, so we can look specifically for these deletions in that first time point and show that this is chromosome 13, so chromosome 13 is indeed mostly clonal, but we also see this subclone that harbors the chromosome 16 and 17 alterations. So, yeah, so basically, what we believe what's happening is there's a branching kind of pattern of evolution where this subclone then eventually acquired more deletions and expanded to become the ascites dissemination. Yeah, but what's cool is that because we're doing this with RNA-seq, now we can directly compare the transcriptomes of these two subclones. And so we can use CDE, differential expression analysis, to find genes that are upregulated in this subclone compared to the second subclone, and show that the second subclone transcriptionally is more similar to the cells of the ascites dissemination. And now that we have these genes, we can, in theory, go through them one by one. We can also do gene set enrichment analysis to show that the genes upregulated and the non-expanding subclone is enriched for certain things like immune system processes, while the subclone that putatively expanded into the ascites dissemination is enriched for things like cell cycle. And I think this is definitely very speculative, but it's possible that, let's say, the subclones are somehow cooperating. Maybe one is suppressing the immune system while the other is allowed to proliferate and grow. But I think this type of single cell approach is really needed to further dissect how subclones may be competing or working together within the same tumor microenvironment. So it looks like we have a few minutes left, but the take-home message really is that I think understanding intratumoral heterogeneity requires data and analyses to be done at the single cell level. Some DNA level information can be effectively inferred from RNA data. I showed with our work with Catherine Wu and Angela and Lili Wang and Cizi Zhang that we have a targeted multiplex RT-qPCR approach that sensitively and accurately detects SNPs and quantifies gene expression, but of course is targeted. So therefore, we had to develop a computational, unbiased approach called honey badger that infers copy number variations and LOHs from single-cell RNA-seq data to enable linking DNA and RNA-level information in a non-biased manner. And I think additional computational and statistical approaches are still needed to really enable this kind of integrative multi-omics analysis and try to understand intratumoral heterogeneity. Yes?
Copy number gains also cause a shifting, but to a lesser extent.
Exactly.
Have you tried incorporating that into your model?
Yes, we have. So exactly as you said, we can see, so if you have a copy number gain, in theory you could see the allelic imbalance in the gained allele as well. But because of the spurious, because allelic imbalance isn't that precise at the single cell level, in order to detect amplifications, it's actually much better to use the expression-based karyotyping, in my opinion.
How does are you familiar with Peru's recent work with pseudogenesis, where they're trying to find copy number variation from blood, right? So circulating
tumor,
and usually it's a very small fraction. So the question is, how low a fraction can you actually get a readout of this copy number variation? And I think the trick is there's something parallel sort of what you're describing, which is that by phasing the data, you you see the bias, but you also know the direction of the bias.
Right.
That helps a lot.
Right, exactly.
Right.
So I know PAGODA and others have been really working on the phasing component. We're essentially phasing by looking at the direction of the bias,
Right.
but we're actually not like actually phasing. But you're so just to get at your question, what is the clonality that we need in order to be able to distinguish tumor from normal? So basically, we're at a 10% clonality. We're starting to do extremely poor in terms of precision. I think I think for sufficiently large alterations, we can handle like a 30% clonality. But my guess is for the circulating tumor cells, you're looking at maybe 1 or 5%.
Yeah, they can go pretty low, but they don't get the rich readout of interpretation.
Yeah,
Functional
yeah.
outside, so.
Yeah, so I think, to really handle a circulating tumor cell type data set, you would probably have to enrich somehow first. But actually, so let me just put up my last slide with, I'd like to thank my mentor, Peter Kharchenko, all our collaborators in the WU Lab and Fluidigm, SGI, Park Lab, and also funding sources. And you can find me online and email me if you want to grab coffee or something. So thank you.
Thank you for the invitation to present here. So I'll talk sort of a combination talk, basically. I'll try to go over some of the approaches, some of the challenges that I think remain in this single-cell analysis field, at least currently. It's hard to predict because it changes every year so much, but I'll try to go over what we're trying to do now. And then I'll present sort of a vignette, I guess, focus on a vignette on analysis of human brains. Okay, so as I'm sure usually I start with kind of an introduction as to why do single-cell analysis and how people approach this, but I'm sure the audience here is very familiar with this. But just to emphasize sort of the amount of progress we had just in the past few years, because it seems like ancient history, but it was just a couple of years ago that this was the prime machine, right? And so we went from this 96-well microfluidic system to, well, I guess plate systems have been used for a while already. They've advanced quite a bit in terms of quality and a little bit in terms of scale. Still have very significant advantages. They're micro-well plates sort of that now taking over the droplet microfluidics that was pioneered here as well. Obviously increased things by another order of magnitude. We got about capability of measuring basically thousands of cells, analyzing thousands of cells, preparing such libraries. I had some movies of our droplet microfluidics. This is the in-drop sort of idea from, or the in-drop version from Alan Klein. Again, the idea is that you basically encode the cellular barcodes that maintain the identity of the cell, of the initial droplet in which the material was produced, the cDNA was produced. You also have unique molecular identifiers here to basically compensate for some of the distortions of the amplification. Analysis of all these sequences has its own problems, and basically there are a lot of interesting questions on how to clean up these sequences, how to account for errors, and so on. And this is fairly low-level work. I won't go into very much. So I did this fun exercise looking at all of this. I keep telling people that the field is growing a lot, and they sort of stare at me blankly. And so so this is the just the CPUs I've used in my computers, just to sort of recapitulate Moore's law. And actually, I never did this by hand. You always see these published papers and figures. You don't know how well this works, but I just put it in from the Wikipedia, and it works. It's really amazing. So then I did the same thing for the single cell studies here and put it on one scale. So obviously, here we're counting transistors, here we're counting cells that we're measuring. And so these are kind of like landmark studies. We're basically starting with sort of early embryo work, Azim Surani's work, where single cell analysis was particularly important because every cell in the embryo is supposedly distinct. Linison paper from 2010 on, I think, had just over 100 cells, ES and mouse CS, and I forget what was the other, another cell line, I think. And slowly working up to this kind of a few thousand cells, again done on Fluidigm C1, then the droplet methods, and then this is not a publication, but basically this is a data set, right, that 10x Genomics put out with million neurons. So I calculated basically we'll exceed the number of transistors by 2030 or something like that. But realistically, nobody's trying to aim at that. And at this point, it seems like there's sort of diminishing returns you get from having, let's say, a million cells versus 100,000 cells. But then again, if you ask me a year before, I would tell you probably you don't need more than 1,000 cells, right? So I'm not going to go out and say anything like that. We'll see. But practically speaking, that means that the volumes of the data we get have grown up considerably. It's quite straightforward to get matrices of 100,000 cells by about 10,000 genes altogether detected across cells. Obviously, if you just represent as a dense matrix, probably not something going to fit in your computer memory. Those kind of considerations alone require more advanced algorithms, so at least more careful treatment of these samples. The other thing that's important, and I think that's what's really going to push things forward, is the fact that technology already became much more widely adopted. A lot of people paying attention to this. So instead of having these one-off data sets where you sort of look for the first time and then try to see what's actually there, now people are doing more systematic studies where you have sort of multi-sample panels, you have patient cohorts that you're actually trying to compare and get some generalized conclusions out of, which I think most of the current methods are not really aimed at doing, right? They're mostly exploratory kind of things. And then finally, as I'm sure you know, because the kind of consortium level efforts to produce large compendiums of these single cell data sets, to have sort of encyclopedia or human single cell atlas, that's the term for the current project or a bunch of efforts. So most likely we'll see essentially very large collections that, again, we'd like the methods to be able to take advantage of and actually make sense of as well to begin with. Yeah, so there's essentially kind of a computational task to all this in terms of how do you optimize your actual code, your actual algorithms, and how do you pick the right approach computationally. There's also a bit of how do you do statistical treatment on such level, especially if you're dealing with these different batches, different individuals, and so on. And I think a lot of these are sort of challenges that we'll have to address over years to come. All right, so I'll jump back a little bit and talk about some of the work we've done previously just to set the background, and then I'll talk about how we're basically moving that forward. So, one thing I want to emphasize, now this is an old picture, but just the sort of the specifics of the single cell measurements, the types of distortions you tend to get there. Essentially, so again, this is one of the initial, I think, Fluidigm measurements, but we're comparing expression levels of genes, of different genes. Every point is a gene in two different cells. And if they were identical, you would see everything along the diagonal, but you basically see this kind of dispersion here. They're over-dispersed compared to bulk data that you typically see. You tend to see outliers once in a while, though with newer protocols it's less and less. You see these streaks along the axis where you see a gene expressed in one cell, but not at all in the other cell. So basically there's prevalence of zeros. Now, again, I think if you have more sensitive methods, and I'll show you some examples, you'll get fewer of these zeros, but it's still quite prominent, so-called dropout events or zero inflation. So the important thing is that if you're measuring a population of cells, the cells will vary in quality. And some of the cells you measure will be very poorly measured, whether because of the lysis conditions, library prep specifics, and so on, and others will be very well covered, and this needs to be balanced out. In general, these types of distortions will cause problems for straightforward methods. Like if you just apply PCA, let's say on the count data and so on. Count data in general is non-Gaussian, so you always have to worry about normality assumptions when you're applying these methods. Noisy cells, again, tend to form outliers that will essentially mask your true biological heterogeneity. I'm using apples for cells here as a metaphor, but what we've been trying to do so far is basically come up with accurate models for cell-specific noise, so cell-specific noise effects, and then try to propagate that forward into population-wide measurements such as variability of, let's say, a given gene across an entire set of cells. So here's some examples. So the model basically we've been using is pretty straightforward. Essentially, we're modeling either kind of a successful RT capture of a gene, and then generation of an amplified, essentially, set of reads. So in this case, this is the expected expression magnitude of a gene. This is what we observed in a given cell. You can think of expected, let's say, if the set of cells is homogeneous, this is the average expression magnitude of a gene in this population. So if we do amplify it and capture it, then we see some set of counts. And that's the blue distribution. And that's essentially just modeled as negative binomial, as you typically do in the bulk data. And then there's this red distribution, which is basically a set of genes that we fail to detect for one reason or another, whether they're not present in the cell, biologically, for stochastic reasons, or because the actual measurement procedure failed to detect them. And so they're distributed into this other sort of failure distribution, which is a low-level kind of Poisson background. The main thing is that we tried to basically measure or model the frequency of these dropouts as a function of true expression magnitude. That gets us a little bit of extra information. So what I wanted to do actually is just show you some of these error models. I think they're quite telling in terms of just diagnostic as to what's going on. So I'll show you a few examples from a few techniques. So this is from fluidic. Again, if you just focus on the central slide here, so this expected expression magnitude, this is what we see in a given cell. And what you can see, first of all, like quite a bit of overdispersion here, right? So the dashed line's a 95% confidence interval, so it's pretty wide. There are also points above here, and this is actually something I looked at extensively, and in fluidic, it's fairly specific to that technique. Essentially, these are genes that for some reason are over-amplified considerably. There are no UMI corrections here, right? This is the 96-well chip. And you can't explain this with negative binomial, and I tried a bunch of other distributions. Essentially, they're kind of outliers, but they occur again and again. The other thing you can see that the genes you do detect, most of them are bunched up in this area. So essentially, the reported magnitude is kind of squashed, right? So that means the dynamic range is pretty poor. There's some extra dropouts on top of this negative binomial, but you already see that it's so wide that it accounts for a lot of zeros to begin with. So here's a different technique. So this is indrop, one of the droplet methods. So here, so this is a UMI-based method. So we reduce some of the amplification and distortions. You can see the overdispersion is narrower. The whole distribution is more elongated, so you get better dynamic range. But you can also see these discrete levels, and these are actually individual read levels, right? So this is a single cell, so it translates to a unique number, and that's because the measurement is very sparse. Because it's sparse, you also have a lot of zeros. Actually, that's a frequency of zeros, up to like 60% or 40% even at some reasonable expression level. So you miss a lot of things, as we expect. But you get fairly linear and a nice kind of amplification. This is my favorite so far, SmartSeq2, but actually there's quite a bit of variability in SmartSeq2 results, let's put it that way. This particular one comes from the Karolinska Institute. They have a facility there, and they automated the whole thing, and I think they came up with a protocol, so somehow it works out pretty nicely. But so what we're looking for is basically, again, nice dynamic range, so it's fairly elongated. The overdispersion is not high. Now this is actually remarkable because there's no molecular identifiers in this data. Please.
Sorry, what's the y-axis in the middle? Because it says counts, but I don't see integers. So.
Well, it's log. Sorry.
Is it log counts?
Yeah.
Great. Okay.
Log 10. Yeah. Right. So there are no UMIs in here, right? So basically, if you whatever effects of over-amplification you have, you see them here, and apparently they're able to actually keep it under control reasonably well. And the amount of dropouts is fairly small. But compared to some of the modern methods, it's relatively low throughput. You get like 384 well plates, and it's very expensive for that. But if you want high quality measurement, that's where I go. Oh, expected. Yeah, it's a little bit tricky. So I gave you an easy answer. If it was a homogeneous set of cells, right, that would be an average expression magnitude of a gene per entire population. But obviously, we're not interested in homogeneous populations. So, in fact, to derive this, what we do is we get sort of the neighbors of that cell. We actually go through a kind of pre-processing of clustering cells, and then we consider the neighborhood to be homogeneous, or at least as homogeneous as we can guess. We basically, because these error models do rely to some extent on expression dependencies, so that's how we get it.
So expected the nearest neighbors.
Yes. Yeah.
Like the most serious median distance.
Correlation distance. Yeah.
So this is the model that's described in the SCD paper or the PAGODA?
It's in PAGODA because SCD didn't consider heterogeneity. So in the second paper we consider heterogeneity, so we do this k-nearest neighbor model fit. Yeah, this is actually it's a good point, and this is something that's not, I guess we haven't emphasized, but it's something you have to do. So even in these newer methods in processing, you can typically get crude populations pretty well, but if you want to refine it, you're better off actually refitting the cell sizes based on the initial subpopulations to get more accurate estimates.
And what you're showing in these plots is the average, so the observed counts. So this is a dot for every gene across the entire data set?
No, so it's a single cell.
It's just an individual.
It's just one cell. The expected magnitude is derived based on, that set of cells that was in the neighborhood, right? So it's not, except for that cell, obviously. So it's sort of averaged across a part of the experiment. But the counts are for the single cell because we're trying to infer parameters for a single cell. All right, so yeah, the thing, and then again, I think in the initial measurements, initial data sets that we had, I think the noise was limiting in a sense. We didn't have a lot of cells, and the measurements were quite noisy, so we kept looking for additional statistical power to actually resolve things. And even now with modern data sets, there are plenty of situations once you kind of drill down into smaller subpopulations or subtle processes, you do need extra statistical power. And so the approach here was to basically bring in some of the kind of prior biological knowledge in the form of very simple gene sets. So that was the level of abstraction I think unfortunately we still are. So the idea is, again, just parallels the gene set kind of analysis, either gene set enrichment or just hypergeometric enrichment analysis.
Can you remind people what that is? What is a gene set?
A gene set. So a gene set can be anything. In fact, something that basically gives you a week prior that these genes can work together, at least some of them can work together to accomplish some biological function. Now, in practical terms, you could take something like Go ontology, right, gene ontology, and then take essentially Go categories and treat them as gene sets. You could derive gene sets in other ways. You could go, for instance, we've done this at some point looking at brain. We went to Allen Brain Atlas database and derived clusters of spatially correlated genes. So it's expression, but it's somewhat unrelated to the exact thing. And it turns out that these genes, obviously, they do correspond to some sort of functions that happen in the brain in a coordinated manner. And giving such gene sets will also be very informative. So you can derive it in a variety of ways. You can add your custom sort of literature-derived things, but we don't actually put restrictions on that. It's a week prior, so in some way, if you put in a wrong gene set, nothing bad is going to happen. If you put too many, you'll get a little bit of a penalty on multiple hypothesis testing. But with so many cells, you typically not, you have some leeway there. Right. So the idea, for these standard testing, let's say in differential expression, if you have two conditions, you run differential expression, and you find that, let's say, none of your genes are actually statistically significant differentially expressed, right? There was a weak perturbation of a sort. But then you take, let's say, a gene set of actin genes, let's say actin pathway, and you find that 4 out of 10 are on the top, let's say, within the top 4. Now, the statistical chance of that is pretty low, right? And so you can actually achieve statistical significance this way. And you get two things. You get sort of increase in power of being able to detect sort of actin-related changes. And also if you have a number of gene sets you're testing, then you get some interpretation hits. So the idea here was, pretty much the same. If you have heterogeneous population of cells, and then in some subset you see all of a sudden these actin genes, all of them go up, right? That tells you two things. That most likely there is actually a subpopulation that is distinguished, that is a real subpopulation, and it's probably distinguished in part by the actin pathway activity. So some statistical bits. So basically why we bother with these error models, right, is because we want to be able to actually normalize and properly account for these types of errors in the downstream analysis. And here the first downstream analysis is sort of normalizing the variance of the gene across the data set, right? So we're looking essentially for genes that are that vary too much within the data set. And why do we do this? If you have, let's say, a simple subpopulation structure with one half expressing the gene highly and the other one not expressing it at all, right? That gene would have average expression level, half of that, and it will have quite a bit of variance. Now, if you compare it to a gene that is evenly across all of the cells expressed at that half level, you would have much lower variance, right? So for the same expression magnitude, which is what here on the x-axis, if you see higher variance, that makes it more likely that the gene is actually marking some kind of a subpopulation. And so that's what we look for. There's a genome-wide trend here. We tried analytical models, but didn't really account for a variety of data sets that we have. So this is a genome-wide trend. This coefficient of variation squared on log scale again. And what we're looking for are points above this line, right? So the genes that show more variability than we expect genome-wide. And what we're looking for actually is, let's say if we have these 3 genes here that are over-dispersed, we're hoping that they come from one pathway, for instance, let's say actin pathway. And in fact, what we're hoping is that if these are cells and these are genes, that these 3 will be expressed in the same set of cells. If they're hypervariable, they're from one pathway, but they're expressed in random collection of cells, that doesn't give us a subpopulation. That just tells us it's a noisy pathway. Please.
Not a random set, but you could very well have, if you have 2 clusters, for example, right?
Yeah, it could be 2.
Genes from both of them, right? And do you see what I mean? Right?
So they could mark two distinct subpopulations.
Yeah, they'd be mixed.
Right. A little bit mixed. Right. So yeah, that's a good point. So what we do essentially to get this, we're looking for coordinated variation, right? Or basically higher than expected by chance and coordinated. So the coordinated part we get through this PCA, weighted PCA, right? So if you just look at the first component, right, you might essentially get, mostly one subpopulation if it's larger and not the other. If you look at the second component, which is an option, then, you could get that second subpopulation. But in practice, there's an option actually to do more than one principal component of each gene set, but we don't do this because it almost never actually gives information, or at least not information that you can derive from other gene sets. Again, a little bit of a technical bit. So, if you look at expression of a single gene across a population, so there are some cells that express that gene and some cells that don't express that gene, or at least we don't detect it there. Now, it could be that essentially it's a distinct subpopulation of cells that just does not express that gene. Or it could be that we've simply failed for technical reasons to detect the gene there. And so because, again, we know these error models estimates, we can sort of estimate what the fraction here of true zeros, the undetected measurements, right, versus true zeros where, cells didn't express it. We don't know which cells they were, but we know the fraction, right? And so this is why we went with this. The variety of strategies you could use to actually make sure your PCA analysis, doesn't recognize that as a separate subpopulation if it's mostly technical failures. We settled on this weighted PCA approach, which essentially weights by the probability of a technical dropout. And the logic was, so again, if this is a single gene set, the cells are columns, the genes are rows, expression is from low blue to high in red. We're looking for cases like this, where there's a bunch of genes in coordinated manner distinguish, let's say, these cells as opposed to all the other cells. And this is captured by this first principal component. Now, if this is a statistically significant trend, right, then this first principal component will explain, more variance than we expect by chance. So that's the test that we're essentially doing. And we're doing this for all the gene sets that are given to the database, let's say all the gene ontology or something else. Now, the annotations, if you're using gene ontology, they're not perfect. They don't capture a lot of the more subtle processes. So we also, not to miss obvious things, we look for essentially correlated clusters of genes in the data and treat them in fairly similar way, except for the actual statistical evaluation of low dispersion. So what you end up with after all this testing, you can put together just the statistically significant patterns, right? And so you end up with a collection of them, and you can see that there's some kind of clustering here. It's a fake example, obviously, but these two will, for instance, separate the same 2 cells, and these 4 will sort of separate these 4 cells on the left. And in reality, this is a very strong trend because a lot of gene sets, let's say gene ontology annotations, are redundant or overlapping. And then if you actually have a distinct subpopulation of cells, it will probably be distinguished by more than one biological process. So even if the genes, if it's different genes actually separating, they're still separating the same subpopulation. So just to enable kind of navigation of this, to kind of line up and arrange your apples, we're basically ordering this, we're clustering these and ordering them. And then the key thing, again, this is exploratory tool, right? The key thing is actually interpreting what this means and looking at the different aspects of variation to figure out what's important to you, to your particular experiment, what's likely to be an artifact, and so on. So we try to basically build interactive tools like a UI that you could explore this and look at specific genes and so on.
So, question?
Please.
So you have on the left, the upper part, sort of supervised gene sets, and the lower part, you said you were looking at correlation of the data to sort of extract gene sets in an unsupervised way.
Yeah.
How do those compare? I mean, do you tend to find more interesting things from one approach to the other? Do they tend to coincide or be totally different?
Yeah, it's actually quite clear, I think. In the very noisy data, if you sort of limit it by statistical power, then the annotated gene sets will win. Just because the prior they bring will actually increase the p-value significantly. Now under it, let's say if you just rank them, like here they're ranked by the z-score, right, or the p-value essentially. So under it somewhere you'll see a cluster, a gene cluster that is sort of captures that same pattern, but you won't have as much statistical power because you sort of picked it up and then it could have happened by chance essentially. But if you have very good quality data, then the clusters will actually win because they're able to discover more extensive patterns. Let's say if you're looking at cell cycle or something like that, it's different in every cell type, right? So it's, if you put in your annotation, it's going to miss half the genes or more. But with a cluster, you're able to capture a much larger set of genes that show this coordinated pattern, and so as a result, you actually overcome this little prior that you had from this gene set. And I think that's actually illustrative, sort of towards the direction where we're going. That I think that we're trying to move away from gene sets into essentially just the kind of prior experiment-based annotations, because I think they actually bring more information than the simple gene sets we're able to manually curate.
Like just more unbiased, data-driven.
Yeah. So I'll show some examples.
And
Please.
one more question. So many times there are cases in the data where you do have a few very distinct clusters, right?
Yeah.
And then you want to drill down into each of these and find the substructures. So would the way to use this method, would that be to restrict, to rerun this on a limited number of cells, or to use more principal components?
Well, no. So we basically advocate rerunning this. So it was actually in the paper, there was a little loop going back. And the reason is this variance normalization. So So the over-dispersion analysis. Now, we don't throw out the genes. We simply renormalize the variance to calculate this residual variance, right? So you don't, like if the gene is not over-dispersed, you don't lose it. You still have that pattern, but it's statistically, it could be downweighted. Now, the problem is if you have, let's say, one, kind of uniform cluster and one heterogeneous cluster, but they're very distinct. Most of the variance you'll capture here will be the distinction between these two and not the little heterogeneity you want to see within it. So when you focus on it, you should be able to detect it better. So you're better off doing this. In the new version, we tried a couple of like automatic recursion, but I think we'll do a different approach. But yeah, you can rerun it. Please.
So in your experience, currently, what's the fraction of sets that don't need prior? What's the fraction of clean sets that you get from single cell?
Data sets.
Yeah.
Well, it's hard to say. It depends who's doing this, right? In my lab. No, I think that obviously technology is getting better, and on average data sets are getting cleaner. I think so. There are two parts to this pathway thing, right? And as I mentioned in the beginning, there's one that's getting statistical power. And I think going forward, with the cleaner data, compared to our prior, the gene set prior anyway, we'll get more statistical power in the actual data. But the other thing they give you is interpretation. I think that's still very important, and the gene sets are still helpful in that. Even if you have this super statistically significant cluster, it's going to take you time to figure out what it means, but if you have a few hints below it saying that same pattern is exhibited by this pathway or this set of pathways, right, it would be helpful. So just from that standpoint. This is just an example. So this is on cortex, mouse cortex data from Linnerson lab, and this is how it looks. So the columns are cells. These are aspects of heterogeneity. So these are basically patterns, clustered patterns, and so if you look through it, and I won't go into the actual browser, you can sort of pick up the pathways that make up each aspect. So here you had a bunch of myelination pathways, immune response pathways for microglia, for instance. You can look at individual genes and search for additional patterns and so on. And then I think in this particular data set, you could actually go through and interpret most of them just by looking at this very easily. But obviously, in a lot of cases, it's harder. Okay, so moving on. So like I mentioned, now we're basically starting to work with data sets that have thousands of cells routinely. 10,000 cell data set is probably more typical now, especially if you run several batches and replicates. Most of them have molecular barcoding, and that helps a lot in terms of simplifying error models.
Can you
please
unpack that?
Molecular barcoding.
What is that?
Okay. So this is going back to Alan Klein protocol slide. I wanted to explain it there, but essentially the idea is that when you're during generation of cDNA somehow, and different protocols do this in different ways, you incorporate two barcodes. One marks the cell, that this came from. So let's say the droplet. Most likely this barcode will be unique for the droplet. But then it also has a little random sequence next to it, let's say 10, 20, or 6 nucleotides that are synthesized to be random. And so you generate the cDNA with that, and then you make sure that in your further processing in second strand generation and amplification and so on, you basically maintain that region. And that means that all the products that will be generated from that initial molecule will carry that 6 nucleotides, so this unique molecular identifier with them. And so if you amplify that particular molecule 20 times, you can look up the unique molecular barcode and see, okay, it's the same gene, same cell, and it has the same molecular barcode. So most likely all these reads came from this one molecule, and I shouldn't count it more than once. So that's the logic.
So
right?
save complete dropout, it gets rid of noise from amplification.
Okay, well, so it doesn't get rid of the noise of amplification. So it limits the impact. Let's put it that way. So in principle, if the barcode is very long and you're sequencing it perfectly, then I think you can get rid of most of it.
You're saying the combinatorial space is quite big enough to prevent overlap?
Right. So it's not purely combinatorial space. It depends how you generate these barcodes, but they're not uniform for the most part.
Okay.
The other issue is that there's sequencing errors associated with them, and so unless you have some strategy for distinguishing them, you might overcount highly amplified transcripts. In fact, if the error was generated early, you can actually look like a distinct molecule because you keep seeing that distinct UMI, a number of times.
So
it's not perfect, but it's pretty good.
If the UMI system was perfect, it would correct perfectly for
yes,
amplification bias, but it's not. The UMI system is.
That's a fair way.
Okay.
Anyway, but it does clean up things considerably in terms of over dispersion.
Okay.
So so that's a now prominent feature. Although I should note, there's no full-length protocol that gives you a full-length transcript, right, with UMIs, which is a pity because we would like to see that. So you get, but the other feature is that they tend to be sparse. And I don't think it's going away. You can get as few as a few hundred molecules per cell. You get a lot of cells, you can get 100,000 cells, but the coverage of individual cell can be pretty low. Now, part of it is protocol, part of it is basically sequencing effort. Even if you had a very good protocol, then sequencing it to a considerable depth might still be a challenge, at least expensive. So those are kind of the features we're dealing with for foreseeable future, let's say for this human single-cell atlas effort. And so this is what we tried to kind of redesign the new system around to make sure we can process data sets like this. So basically a lot of this was just optimizations, kind of simplified residual variance estimation and corrections. The idea is to implement very fast clustering, visualization, and differential expression, and so on. Still keep the whole idea of kind of alternate cross-cutting aspects and pathway analysis. So this is actually a video that ran in real time, and this is a data set from 10x, I think, the PBMCs, 33,000 cells. So just some simple filtering, residual variance estimation, clustering, and then some visualization. So about 20 seconds. So now, obviously, you don't need it like this. If you want to run pathway analysis, it will take a little bit more time. But it kind of gives us a platform to start building more advanced things going forward. So yeah, the UI. So actually, this thing is we just put it up there, but it's really an alpha version, to be honest. And to a point, so we're building a new UI that will be scalable. But right now, it's already there. But right now, we also have the use of the old UI. Turns out UI actually scales to 10,000, 20,000 cells reasonably well, unlike the rest of the backend. So yeah, so you could try to check it out, but probably in a month it's going to be a lot. We're going to have tutorials up and so on. Okay, so what I wanted to do, actually, just since it's a, I guess, technical seminar, to give you some idea about some of the solutions we actually used, and hopefully you'll find them useful as well, for sort of a standard pipeline. Now, this whole thing is built to be flexible in terms of how you exactly process data, what sequence of steps, but here the pipeline, I'll show you sort of a classic where you do some filtering, some censoring, some cell size estimation, PCA, and then we go into graph space to do both clustering and visualization, and then differential expression. So yeah, PCA. So again, we're dealing with very large sparse matrices, right? So they can contain a million cells, but they're very sparse, and that actually gives us a big advantage. So what we're using is this Langkos iteratively restarted algorithm. The idea, again, it's not our method, but it's fascinating how all this works. And the idea is pretty simple, actually. If you have this matrix and you're trying to find the first eigenvector, you can just take a random vector x and then keep multiplying, by a matrix, and with some normalization steps. And in principle, it should converge to the first eigenvector. Now, this is not really a practical approach. It's unstable. But if you look at the actual space of these results, right, the initial vector, the vector multiplied once by the matrix twice, and so on, this is formally known as Krylov space. So actually, you could analyze this entire space to arrive to converge on the solution much faster. And then, so you do this, you essentially find these vectors, eigenvectors, in this Krylov space called Arnoldi vectors. You have to do some restarts to make sure that it's stable. But the whole thing is very fast. There's an implementation in R with IRL BA package. I recommend it highly.
What's the computational complexity in
Sorry.
terms of the?
Wow.
Can you speak to the computational complexity in terms of both the sort of rows, columns, but also just how many eigenvectors that you want to get out?
Yeah. Good question. So now the complexity of this is actually it's hard to answer because it depends on the sparsity. Okay. Yeah. So depending on how.
Are you doing sparse matrix vector multiplies? Is that?
It's all sparse. Yes.
Okay.
But also the convergence of the algorithm depends on basically being able to find blocks within the matrix that.
Right. So sparsity of the spectrum.
Yes.
Like
yes.
Separation. Of the
spectrum. Right. Which is hard to assess, from just looking at the data.
Right.
Yeah. In terms of the need for the eigenvectors, it varies. I think you can get away. Again, depends on the sort of the level of detail you want to do. Usually after 20 or so, you start repeating. Sort of getting things that are not really making a difference to the final result, at least as far as clustering is concerned. I think a lot of times go deeper because it doesn't seem to hurt, but I think for the most data that we looked at, that would be sort of the order of magnitude.
Speaking, do you find that the time is proportional or quadratic in
the number of...
Oh, the number component, it's linear because it's the same, it just iteratively calculates itself.
Does the quality go down as a result of, or is it fine? Like how deep in the spectrum?
It's approximate, right? So
Does it get worse as you go deeper in the spectrum?
I'm sure it does. Actually, we didn't do this test.
We're worrying about something else. That's why we're asking.
I see.
Yeah.
Well, it's pretty easy to do. I mean, through simulations. I'm sure it's an approximate method, so
You can check this.
the ability to resolve it clearly would go down for the lowest one.
And the sparsity is typically about 1 in 100 order of magnitude for these matrices, the number of fraction of non-zero. Is there any rough range of order of magnitude for sparsity that you've seen?
Well, you're filtering to genes that are...
So after QC.
Yeah, we do this filtering, but that's what I'm trying to figure out. So, let's say we get about 15,000 genes, and, but then you have a distribution of molecular sizes.
Yeah.
A lot of them, can have, let's say, 1,000 genes on average, right? Some will have 300, some will have, 4,000 or 5,000. So that's sort of...
Those are cells. Some cells.
Some cells, it's in the genes. So that tells you about sparsity a little bit.
Right. That's true. So it's more like 10% to 50%.
Yeah, something like that. It depends on the data set again.
Sorry, of non-sparse, of non-zeros.
So I'll show you some data that will be much sparser than this.
Yeah.
Okay. So the other thing, like I mentioned, we go into the graph space, but to go into graph space, we need to basically find, let's say it's a k-nearest neighbor graph, and so we need to find the nearest neighbors, right? If you have a million cells, then there are a lot of sort of possibilities to check. We cannot calculate the n by n matrix, right, of distances. So again, this is a very popular problem. There are many methods there, but I just wanted to emphasize, the one we've ended up using because it was an order of magnitude faster than the next best thing, and we were trying fairly advanced things like locale-sensitive hashing and so on. So so the idea is based on this small world graphs, which I used to do metabolic networks analysis at some point, and so I still remember this a little bit, but I didn't think they were very useful, but here apparently they are. So the idea is very simple. Like if you're trying to navigate a small world network, you can actually do this very efficiently, right? So the idea here is to incorporate, in this case, cells into these layers, multiple layers, where your hubs of your network will be sort of on the top, and then less hubby nodes will be a little bit lower, and so on. So when you're trying to, let's say, go somewhere, some small town in Europe, right, you'll probably be flying to Frankfurt first, and then from there, going somewhere else, and then taking a car, and so on. You already know the route because it's sort of easy to figure this out. And so again, if we place an arbitrary point here, a query point can be one of our cells, could be something else. We can easily find the closest hub, and then from there find the next closest one, and so on. So this is an algorithm that was published just last year, and again, it's based on the initial compilation of the structure, right? So you sort of insert your cells one by one, and there's a heuristic that allows you to pick the right level for the cell. And then after you compile this engine, so to speak, you can actually query it very fast for nearest neighbors of a given cell.
Is that conceptually very different than a KD tree? Like something that sort of partitions into these larger...
I think
it's pretty different.
Right. No. So I think that on one hand it's a sort of a tree decision anyway, right?
Right.
There are extra heuristics when moving between some of these nodes, but I think the key thing is how they lay it out in layers. So the heuristic of basically determining sort of the multiplicity branches at each level, which is essentially what this layer density will give you. I think that's where the trick is. And again, it's a trick that is specific to, that works, for instance, for this system. Because if you look at, let's say, correlation distance or other distance, between the cells, it will look like, a small world network. Another network might not, and this method wouldn't work so well for it, right? But, in this data we're analyzing, it's very much along the lines here.
Is this exact or approximate?
Approximate.
So if I understand this model correctly, so the underlying assumption is that even though you're using the layers, the distance you'll get is essentially equivalent to the distance the nearest neighbors you'd get without.
It's approximate. I mean, at the end, yes. The distance you can always reevaluate once you have the neighbor, right? I mean, it's easier.
No, I meant the number of
Yeah.
nodes you need to go through to get to
Well, the number will be a lot smaller, but at the end you should get very similar set of neighbors.
Yeah.
Okay, visualization also. So this is just visualization PCA space. Please.
Sorry, maybe I forgot this, but in which space are you doing the calculation of the nearest neighbor? Is it in some PC space or the original?
Yeah, it's a good question. So with the pipeline, you could do it in either space. In the workflow I showed you, we're doing it in PC space. That's why we did PCA first.
But with lots of PCs.
Consider sort of alternatives to that space itself, which is maybe using like multiple kernels or.
No, we haven't used. Actually, it's the second time I get asked that question. I guess I should look into it. But we considered alternatives. I mean, first of all, you could use entire set, as it is to get some transcriptome-wise distance measures, and on smaller data sets it works fine. We've tried things like random projections and so on. I haven't tried kernels, to be honest. Again, I think the idea was to make it really fast, and given how fast this PCA was, we figured we could do this. But yeah, good point. So there should be. So we have this in the concept in the implementation actually is of reductions. So you have the full data set and you can generate a bunch of reductions, whatever they mean. So one of them could be PCA, another could be something else.
So I have no way. I hate to prove this point, but in my experience, I think it's the reduction which happens to be the secret sauce. And I don't know what the right reduction is. Okay. It seems to be downhill from there.
Yeah, we need benchmarks.
Right. You could think of PCA as a regularization a little bit, and not just keeping only the spectrum, and not just computational efficiency, right?
And if you did some kind of spectral reduction, like diffusion maps or something like this, you could perhaps get a better metric to then
adapted to the shape point.
Right. But yes, benchmarks.
I think benchmarks and time.
Yes.
For implementation, or at least participation from others. It's an open source effort, so everybody's willing to pitch. Okay, so visualization. So PCA used to work quite well actually for visualizing things. It wasn't a method of choice, but used to work. But now, with so many cells, you have no hope. So this is what PCA looks like, and you see the numbers here. These are different clusters you actually have, and so if you visualize it with another method, that will be better. So you could at least view it. Now TSNE is a method of choice.
Will not be named.
Sorry. No, it's a method of choice. It's also if you try to run it on 100,000 cells, it's going to take some time, and so we wanted to move away from this and so the approach we're using, it's this is an existing method called large-viz. It's very essentially the rationale is more or less the same, but it's very aggressive in terms of actually considering which distances to optimize during these iterations, which ones are not. And it's a little bit less stable than TSNE, to be honest. There are like a few knobs and whistles there to tune, but generally speaking, with a reasonable set of parameters, it's able to visualize clusters reasonably well. I think the only concern we had is it tends to stretch out some of these clusters, but I think this has to do with the PCA, with the embedding, right? Sorry, with the reduction. Please? Right, so the complexity, again, there's no guarantees here because it depends on the sparsity of the graph and so on. Essentially, in our testing, it turned out to be close to linear, which is considerably better. I think. Yeah. So it's n log n, right?
Yeah. TSNE has its Barnes-Hart implementation, which is n log n.
Right. Yeah, that's what we were benchmarking against.
Right.
And they also have there's like a multi-core version of that, which is just very often it's not actually the complexity, but the implementation these days. Right? So just somebody writing the code again properly in C is with proper cache coherency,
sure.
Right? Is a game changer, right? So somebody did that sort of.
I mean, the constants matter is another way to put it.
Yeah, constants matter.
Sort of like
The constants matter a lot. Yeah.
Again, I think if we had a faster TSNE, I'd probably use it. Sorry. Okay. We'll check it out.
Okay.
Again, I think in terms of the PAGODA 2, there's simply sort of kind of generate embedding function, and it has multiple options. And so right now it's TSNE and this large viz, and if there's a faster TSNE, we can add that.
Just note, the size of the clusters is also doesn't have any interpretation in t-SNE either. Two isolated clusters have relative sizes that have almost no interpretation. So
yeah, I mean, I think all the same.
has these disadvantages as well. So.
Right, but all these limitations stay here.
It doesn't overcome any of those.
No, it's in principle essentially trying to do similar things, just much faster. Okay, right, so this is a new point. So this is basically us trying to move away from gene sets. And how much time do I have? 10 minutes?
Yeah,
5 minutes? Okay. So not a lot of brain data, I guess I'll be able to show you. But
12 minutes.
Okay, maybe a little bit. So again, moving away from, as I mentioned, I think of these gene sets as very weak prior, biological prior. And so now that we're getting all these nice reference sets, even within our own lab, within the studies we're doing, it's obvious that we can take one data set, and then the next time we get a data set, let's say from another patient or something like that, we're better off interpreting it using the information we were able to actually glean and annotate on the first data set. And so we'd like to basically use things, existing data sets as references. And there are a couple of ways in which you could use it. So, well, first of all, this is kind of the difference between analyzing everything from scratch versus bringing in some kind of reference information. But if you bring in reference information, you could do it in two ways. So you could just essentially figure out what are the sort of cell types and patterns, and just, let's say, classify your new cells, your new data set based on that. So essentially, then you're looking only at the reference information, nothing else. Now, what you typically want to do is to capture both. If there's a known kind of a pattern or cell type and so on that you've seen before, you'd like to capture that and be able to separate it clearly. But at the same time, if the data set brings you something new, a different cell type, that you haven't seen before, you want to make sure that it's clearly separated and distinguished instead of being lumped in together with something else. So, for us, we're using this kind of reference segmented approach, essentially, where we try to incorporate the essentially axis of variation that we've seen before, that distinguished cell types before, let's say different immune cells, and then incorporate them on top of the sort of axis of variation that we find in the actual data. So this is an example. It's a set of actually a pool of 3 different prostate cancer metastasis samples, and so this looks like, it looks more or less the same, but this is actually based on this kind of augmented space. So what I've done here is I've taken a blood reference, so PBMC reference, and incorporated it on top of the its own sort of sample reference axis of variation. Now you get some clusters. Now because you have this reference, you can actually try to project the reference clusters back and see what they were annotated as, right? So that gives you the hints, right? So again, the logic is the same, right? You get some hints, and you also get increase in statistical power, because if, let's say, the blood sample separated B cells clearly, right, then we actually have already sort of an axis that allows us to separate B cells, and most likely they'll get pulled away from the rest of the distribution, even if there are not a lot of them. Okay, now you notice that these 3 clusters, for instance, did not get any kind of reference mapping. In fact, I mentioned there are 3 fractions here, and 2 of them have mostly hematopoietic cells, and this one has mostly tumor cells and stroma and so on. So, and actually it should be in blue, so these are the blue clusters. So basically, the advantage is, if you do a purely reference-based analysis, then these clusters would get, either lumped or get positioned very close to some of the hematopoietic clusters, although they're clearly distinct. Please.
For reference, you can only use single cell data that are from the same technique or similar technique from the same lab, or you can just mix and match.
So that's a challenge, and I honestly I don't have an answer for you at the moment. I mean, I think right now we're trying with different labs, same technique. So there are 2 parts to this. Actually, one is easier than the other. First of all, these, I keep saying that they're sort of aspects of variation. Essentially, they're sort of robustified versions of the principal components we pick up that actually distinguish the clusters. We try to trim it so they are more robust, in being moved over to a different data set. But so there are 2 parts to this. One, as I said, you're getting extra statistical power because you're adding some kind of an axis that separates things. Now that's actually, it doesn't have a lot of responsibility, that particular part, because as long as you kind of get the direction right, it will probably separate things correctly, or at least help you separate it. The other is mapping the reference. Now that's more challenging because you have batch effects, shifts, and so on. So we're still trying to optimize this. I mean, I think they're meant to be hints, and so I'm not too worried about this, right? They're not sort of ironclad annotations, but it is a concern. I think we'll have to see. How we also don't have as much data as we'd like to test this on. So I think going forward we'll try to figure this out. All right, a little bit about the brain. So this is study's collaboration with Kun Zhang at CSD and Gerald Shun, who's at Scripps. So the idea is to use single-cell measurements to look at the human brain, at heterogeneity within human brain. We're looking at essentially a couple of regions, the visual cortex and the frontal cortex. It's all nuclei-based measurements. So the technique here for the actual expression measurement for RNA measurement is essentially drop-seq, but it's a nuclei drop-seq. The reason it's nuclei because the brain's, neurons and actually brain tissue is very difficult to dissociate in a viable condition. So you're better off essentially extracting nuclei, breaking down the cells, and just working with the nuclei. Now, obviously, you get only nuclear transcriptome, which can be limiting, and we did some studies on comparing them, but it's difficult to generalize across different cell types.
That might be a little confusing.
Please.
That the nuclear transcriptome is not the transcriptome, because presumably the RNA comes from the DNA. So what is the difference?
So you have some molecules, some mRNA molecules that hang out in the nucleus. Those that were just produced, being produced, those that came back, those that never left. So you have some collection there. Now, strictly speaking, we basically, from sort of biology textbook, you would expect most mature mRNAs to be in the cytoplasm. And so if you come in, let's say, with a poly-T primer to try to fish for mature RNAs in the nucleus, you shouldn't get much. But turns out you get quite a bit. Now, it's obviously a biased sample. I'm sure there's some transcripts that are policed very well and sit entirely in the cytoplasm. And it's also probably biased towards things that are currently transcribing and so on. But again, what we're trying to do is classify cells, and given how big the actual measurement space is, we can do reasonably well. Okay, so this is visual cortex with 16,000 cells. So we're basically classifying different types. Again, there's a lot of inhibitory neurons, excitatory neurons, oligodendrocytes, microglia here, astrocytes, endocytes, pericytes, so endothelial cells and pericytes. So we can look at the markers that's typical for some of these cell types. Now, obviously, like these different inhibitory subtypes, would be very difficult to distinguish. We're just kind of coming up with a classification, and we don't know what to look for. A little bit was done previously on C1 platform. So it's a previous publication from Kun Zhang as well. And so we could just compare some of the groups that were classified there in this previous work using the C1 platform and the ones we're picking up here. So in the drop-seq, we do pick up more. This is also frontal cortex measurement here. I'll show you in a second. So a little bit less data, 9,000 cells. We don't pick up all of the subpopulations here. Some of them probably due to sensitivity. Others seem to be lacking, some of the excitatory subpopulations. Now, so the other thing that was done in this study is epigenetic measurement. So essentially, it's kind of like single-cell chromatin accessibility assay, kind of like single-cell ATAC-seq. They have a slightly different protocol, THS-seq, which is based on this hyperactive transposase mutant that actually gives us more yield per cell. But the approach is based on this index sorting, so like Jay Schindler's lab, split pool indexing. So essentially you sort the nuclei, you label them through transposase reaction, and then you actually relabel them with a PCR. So essentially at the end we get nuclei measurements again, but of chromatin accessibility, right? So we get reads in positions where and the nucleus was able to access the DNA and make the cut, right? And obviously there are a lot of genome positions where you could potentially make a cut. So we focus on peak positions, which we get from pooled data. So pooling across all the cells, just looking at positions where there was a a statistical increase in the number of actual reads. So this allows us to get sort of signal per cell, kind of like we have genes, per cell in transcriptional measurements. Here we have reads at peak positions, usually enhancers or promoters per cell. There are usually a lot more of them than genes. Now, the other thing to consider here, this is data has, well, it's very sparse. We get about 10,000, let's say, reads per cell. And it's also compressed. So if you think like the most accessible site in the chromatin will still get 1 or 2 reads at most in this assay. So when we're modeling this, we have to use a different model instead of this, whole negative binomial mixture and so on. Essentially, we used a right-centered Poisson model that allows us to limit some of these accessibility effects. The other thing that was important here is a size estimation. So there's the size factors we talked about. So here, because you're using different model, you need to fit it in a different way. And then after you do this, you can basically, from the Poisson, the centered Poisson model, you can use deviance residuals to go forward in fairly same way. And so, yeah, this is just illustration of the data, but I'll tell you about the key thing we have to do basically is match up transcriptional populations with expression populations, right? Because our ability, this is the chromatin data, right? So this is, let's say, a cluster here, and we've been able to annotate this astrocytes, but it's actually very difficult to do because, we don't have good annotation for enhancers, for instance. So the approach that we took essentially is taking transcriptional data, looking for transcriptional signatures of a given cell type, and translating it into a probabilistic prediction of which sites, which, let's say, enhancers, promoters, we expect to be differentially accessible. And if you do this for one site, your ability to do this is very poor, right? We don't have a good idea of how things are connected, a very loose idea. But obviously, if you have a lot of sites, a lot of genes changing, and then you translate it into predictions of a lot of sites, and altogether, they give you a fairly confident signature for the cell type. So we have this gene actually came up with this kind of recursive method for doing this, for separating, let's say, inhibitory excitatory neurons, non-neurons, and mapping to accessibility from transcription to accessibility. So I'll skip this, but essentially allows us to get down not to the lowest level of transcriptional classification, but pretty low. So distinguish some of the excitatory, some of the inhibitory subtypes. And the reason why we're trying to do this is because we're trying to get this. We're trying to get essentially reconstruct the epigenetic profile, right, the accessibility profile for cell types that we're not able to isolate. And with that, we can do interesting things like look for transcription factors that are actually perhaps driving these cells and look for relevance of these cells in disease. So I'll have to stop here, unfortunately.
Thank you so much.
Thank you.